Hello, thank you all for joining us for today's ISHR-JMCC cardiovascular webinar with an editor's choice selection for June. Just a few quick announcements before we get started. Just a reminder that the JMCC is actively encouraging and welcoming submissions on a number of topics, including metabolism physiology, genomics, genetics, EC coupling, vascular biology, bioengineering, and signaling. JMCC is proud to announce a very fast review time, including just a 4-day decision without review and up to a 28-day decision after first review. A reminder that there are no page fees for publishing in the JMCC. And one of our really most proudest achievements is that we highlight a number of papers, including today's editor's choice selection through the JMCC-ISHR Webinar series. So please check out the website for the journal and also follow us on social media for the latest from the JMCC. In addition to this webinar series, we also sponsor the JMCC-ISHR Mid-Career Investigator webinar series. The next edition of this webinar will be on September 8th. I will post this registration link in the chat, so please sign up for this webinar as well. So with that, I will turn things over to our moderator for today's session, Dr. Emma Robinson, to introduce our speaker and get us started. Thank you all again for joining us.
Thanks, Ron, and welcome everybody to this morning, this afternoon, this evening. I'm not sure wherever you are in the world. And it's my pleasure to introduce today's speaker, who is Sangita Choudhury. She's currently an assistant professor at Boston Children's Hospital, linked with Harvard University. And Sangita's had a really amazing international training. So she did her bachelor's and master's in microbiology in India, followed by a PhD in cell biology at Greifswald University in Germany. She then did her first postdoc at Beth Israel Deaconess Medical Center, and followed by a short postdoc at the University of Tennessee, and has been at Boston Children's Hospital for the last 11 years or so as a postdoc, and then moved her way up to instructor and assistant professor. So it's my pleasure to introduce her talk today. She's going to tell us about some single nuclear isolation technique called SONIC, which can be used for next generation sequencing. And as Ron mentioned, her paper is editor's choice of JMCC. And Sangita is also recently published in some really impressive journals like Nature Aging, Developmental Cell, and Science Translational Medicine. So just everybody, please, this should be interactive, so the questions will be at the end of the webinar. Please post your questions, during the webinar as you think of them in the Q&A box, and then either Sangita and or myself will take the questions and answer them at the end of the webinar. Okay, with that, hopefully I've remembered everything. I'll turn it over to Sangita. Thank you.
Thank you so much, Emma, for this kind introduction. And thank you, Ron, for, orienting me how this will go through. I'm going to get started. I'm going to share my screen, and I will ask if you can see my screen. Can somebody tell me if you can see my screen?
Yeah, that looks good, Sangita.
That's wonderful. Thank you. So thank you, Emma. Thank you, Ron. And I want to start thanking all the JMCC editorial board and Rong Tian, the editor-in-chief, for selecting our method paper as an editor's choice. And okay. I cannot move. Okay, let me see if I can move. Okay, yeah. So as Emma said that, I am going to talk about how to isolate high quality nuclei from the human heart for the single cell studies. And this paper, I will tell you the backstory of this paper before I get into the method. So Sarah and me started my lab 2021 during the pandemic, and we were trying to isolate the cardiomyocyte nuclei from the Bergman's paper. And we were having hard time to get it, successfully amplified. And Sarah and me tried multiple methods. And when Sarah's time was ending, she said, Sangita, why don't we put this method paper together? Everybody asked us So I am really thankful for Sarah to put this paper together. And it looks like, people are really enjoying our paper. After we published, we got a lot of emails and, they wanted to ask more details about that. So thank you, Sarah, to put this together. And Sarah is now, started her new journey as an MD PhD candidate in Princeton and Rutgers. So it looks like, this motivated her to, choose the program. And now to little bit about why we did this study. So my lab actually focuses on understanding the individuality and the diversity within the cell. And especially I am interested in cardiovascular disease. So I wanted to understand the diversity in the heart muscle cell during the cardiac aging and disease progression. So to do that, our lab, it does a lot of technology and tool innovation, and I am mainly interested in, doing the single cell whole genome amplification. Our lab, along with the postdoc, we established a lot of amplification method, to understand the somatic mutation burden in human heart. And of course, we do RNA-seq and the spatial and all of this. So when we do all of this very sophisticated method, the starting material is the human heart. So as I said, my lab studies the aging. And for the aging, this is not a surgical discarded tissue or explanted tissue, it's a non-disease heart. Where do you get the non-disease heart? So when I started my lab, I looked into this neurobiobank tissue. So there you have, of course, diseased heart or the diseased tissue, but you also have non-diseased tissue. Here we term the control if that case or the donor didn't have any cardiovascular related disease on that case. So we looked at this, and as you see that, we have the age, we have postmortem interval and REAN and everything, but this REAN is not heart tissue specific. It's because it's a brain bank, it's a brain specific REAN. So once we started getting this tissue, we had to make sure that, our starting material is good to do all of this very expensive study. So what we did, we just looked at because we got the REAN, so this REAN actually is not cardiac specific REAN, heart specific REAN. It was the brain bank REAN number. And I think mostly it was for the brain. What we did, we looked at the correlation between the RNA integrity number and the postmortem interval. And we do see that we don't see any correlation between this with increased age or with increase in the postmortem interval in this REAN. We also looked at the REAN number with the Afro-American and the white, as well as in the male and female, and we don't see any correlation with that. We were very happy with that. Oh, so it doesn't matter, the older heart is as good heart tissue with the younger heart tissue, or even if the postmortem interval was over 30 hours, the REAN didn't change. So from the literature, if you are selecting tissue, you look at the REAN number. So we are very excited. But then, my lab is particularly interested in the DNA. So I thought, let's look at the DNA integrity number, because that's what I'm going to amplify. So when we took the same heart tissue and we looked at we isolated the DNA just by the chiaxial bulk DNA isolation, what we see that in the donor with the increased age, we see a decrease in the DNA integrity number. Same with the increase, the postmortem interval, we see a decrease. But again, we see that all of these hundreds of collected tissue, the DIN didn't go below 7. Most of the time, the DIN was above 7. So that's what is our, if the study is incomplete in a way, we didn't look, that if the DIN is very low, but I again believe the way the tissue was preserved in the neurobiobank, the DIN was pretty acceptable. And we also compare between the races and the gender. We don't see with any correlation with the DNA integrity number. So now we wanted to make sure, because eventually from this aging cohort, I will move to the disease cohort. We wanted to see that and if this control heart or the disease heart has any correlation with the RIN and DIN, and we do not see. So this is wonderful because, we can use this tissue, heart tissue, and compare between control and disease, because that's what we will do eventually. So once we know that the RIN and the DIN is acceptable, we want to move forward and we look at this DNA integrity by doing the DNA isolation. And what I want to show that if you do not have the tape station, or you have to go to core, you don't have to do on the upper panel. You can just run the bulk DNA on a agarose gel, and you can see that if you see this smear, that means the DNA is degraded. Because we are a genomics lab, our lab use all sophisticated technology and tools. Here on the left, I'm showing you how a non-degraded DNA should look at the tape station. And then on this here, in the middle panel, what you are seeing, a case of the heart DNA where you see a lot of degraded DNA. And here on the rightmost panel with all this colorful peak, what I'm showing you actually, the DIN, or the DNA, degrading DNA, or the perfect DNA peak. So what you see that if you see this big smear, that means you have a degrading DNA. And a single peak shows a perfect DNA. And what we see from the same cases, just take 1 to 7, 8. In this case, this green peak, the heart DNA is perfect, but what you see in the liver, the purple has a little bit of degradation. So even from the same donor, the tissue could have different DNA integrity numbers. So it's very important when you are collecting the tissue from a bank and you are seeing a RIN or DIN, you are thinking, oh, this is really great. But what, our study shows that the bank might not be checking the DIN or RIN for every tissue. So we need to, in the lab, before we initiate your very expensive experiment, it's important that you check the DNA integrity for your own tissue. So once we know in the lab that, okay, this tissue is good enough and we don't see that much of degradation, so then we look at the tissue architecture. And this is a human heart cardiomyocytes section. And what we see that with alpha-actinin, we are staining the cardiomyocyte, and DAPI with the green, the nucleus. And what we show that it's pretty intact myocardium. So this is the tissue, this is the amount of tissue. Usually we get it from the neurobiobank. We make a section, we do this cryo sectioning under the cryo machine, and we get a small tissue. You can start, so this day we were doing the DNA and RNA isolation. So we started with about 100 milligram, but we have done as small as 10 milligram, and you could get, enough amount of nuclei. And once you cut this tissue, you put this tissue in oxygenated cardiomyocyte isolation buffer. I'm not going to read the buffer ingredients, but it's in the paper. And if you want to discuss, I can talk about this more. But once you put you do not make very thin section, because if you want to isolate the cardiomyocytes intact, you have to keep it chunky so that the cell falls off the tissue slowly, with the collagenase degradation. And you do after the test, so every 10 minutes you collect the amount of the cells are getting isolated from the tissue, and you centrifuge in a 300G for 2 minutes, not too long. And you get a soft palette, and we look under the microscope from the for the isolation. And if we see that, we have a single cell, we are very happy. But if we need to digest a little more, you can do some digestion. And here, this is just the cell bright dye. You are seeing that the intact cardiomyocytes, and they are pretty isolated. They are not together. They are not clumped together. This is what you want to see. And this is also, we are showing that from this neurobiobank collected tissue, and these are sometimes frozen for like over 10, 15 years. And we can still see the connection 43, the gap junction intact there. So this is only, so if you are isolating cardiomyocyte from the frozen human heart, This is not to go back and do your functional study. This is just to, if you are going to look at the expression of the bioimmunostaining or the protein, this is what you can do. We have not done any functional study, but I do not know if any lab has ever done any functional study isolated from this frozen human heart. So once we know that, yes, the tissue is intact, we can get beautiful cells, intact cardiomyocytes from there, then we move forward for the nuclear isolation. So for nuclear isolation, really, we need a preparation a half day before. And we make sure that we have all the things we need, of course, the reagents and the small tools we need. If you do not have in the lab many of them, you can, you can see in our paper, the catalog numbers are over there because it's human heart and we do genome studies. So we make sure we use disposable forceps and the scalpel because you don't want to touch your tissue and contaminate your tissue with the DNA, which you do not want to see it. And we also use disposable homogenizer tips so that, we do not carry over any DNA from a previous cell. And I think my life was saved by this 5 ml Effen drop tube because we have very small amount of tissue. And this tip needs to fit inside this Effen drop tube. You don't want a huge volume because our volume is like 1 milliliter. So this was really helpful to fit in this. And and these filters fits into this 50 ml tubes, which are very helpful. And this is the actually insert in this ultracentrifuge tube. And you make sure that you cool your ultracentrifuge and both the rotors. That is important. And you prepared the lysis buffer and everything we did because we are going to amplify DNA and we are going to do RNA work. So we make sure all of our buffers are sterile and we do sterile filtration after making this lysis buffer. And we pre-coat our tubes with the 1% BSA because you do not want your nucleus stick to this wall because it's very little amount of nucleus you're going to get. And if you do not pre-coat your tubes where you are going to centrifuge, it's going to stick to the wall of the tube. And then we also make this sucrose buffer very fresh in the morning, the day of isolation, and the nucleus storage buffer as well as. And all the details to make the buffer, everything is in the paper, but if you need more details, you can always reach out to me or Sarah. So this is again how we start our isolation. We take a small piece and then here we are showing that it's a 1 ml and the tip goes inside and you centrifuge, you homogenize, but only few pulse, not too much because you don't want your nucleus to be damaged. You just want the tissue to be, the cells to come out of the tissue. And actually you are taking your nucleus out of your cells by doing this downsir. So it depends, how hard you are doing, but in our hand, we need 40 to 60 stroke to get the nucleus out of this cell. And in between, we go and check under the microscope if the cells are fragmented. So for cell isolation, you want to make sure your cell is intact, but for nucleus isolation, you want to make sure most of your cells are fragmented so that, the nucleus could come out, but you don't want to be so harsh that the nucleus is damaged by doing this mechanical homogenization. So once you see that over 50% of your cells are broken, then you are ready to do your centrifugation. So then after this step, we filter through the 100 micron and 70 micron, and we again do a very low speed 700 G 10 minutes centrifugation to get all the things palleted. And after we get the palette, we overlay this cell and nuclei palette on a 2.1 molar sucrose gradient. And this method actually was adapted from the Bergsman method, but what different we did that this palette we dissolve in the isolation buffer so that in the gradient we could capture this debris in the middle instead of getting everything at the bottom. So we still get a lot of debris, which I will show you in our facts plot, but this is still better. We have done all the different method, the 10x suggested, everybody suggested, but this is the method where we could get most of the nucleus and little bit of still debris in there. And this is our facts plot. As I said, there are still a lot of debris, and everybody in whoever is working with the heart tissue, they know how much debris we get, when we try to get the nucleus. So it's very important to set your get correctly. And so it took us, I think, maybe 10 trials to get what are our nucleus, which population we really want to sort. But the beauty of the cardiomyocyte is that it's the cardiomyocytes are also polyploid nucleus. So you see this, the separation between all the debris and the diploid, and then you see also see a polyploid population there. And this is the 2N population where you have all the cell type in the heart. And we try to get rid of a lot of debris here, as you are seeing out, a very small, we are very, very strict about this gating. And you also see a peak with the tetraploid, and sometimes we see even higher ploidy. So once we come to this stage, now we have to decide what we want to do, what is the plan for that day. So this is what Sarah beautifully, made this sketch. So we get the tissue, we make sure that we do the cell dissociation, we make sure the DIN is correct, that we do not see the DNA degradation in the tissue. Then we perform this sonic and we come to this for the FACS sort. On that day, we can do both. We can go ahead and do our RNA-seq experiment. At the same time, we can also take this nuclei and we can do a single cell, we can sort those cells and we can do a single cell whole genome amplification. And this single cell whole genome amplification, after that, we do a whole genome sequencing. And the whole genome sequencing, as you know, it's very expensive. We put a lot of effort to choose the best cell. And this is where we put our lot of effort to identify the nucleus which has amplified well and the good quality of this. So now I'm going to go and tell you what we do to choose this perfect nuclei where, we could capture the changes of the genome in this nucleus. So what we do after we do the whole genome amplification, we do our in-house QC before we send out for sequencing. We chose these 4 chromosomes and we do a multiplex PCR and we run in this, it's again an agarose gel if you do not have the kayak cell in the lab, but it's a capillary gel electrophoresis basically. But the beauty of this, I could run all my plate in one batch. And I could see that, which cell has all the genome has amplified. We tried to do more of those chromosomes to see how, but this 4 fits perfectly and we don't see a lot of cross-contamination in there. So so we were very happy with this multiplex PCR with the 4 of those chromosomes. And we use capillary gel electrophoresis by this kayak cell, but if in the lab, you could also do the plain agarose, but it will be like so much work to, choose your best cell from this 100 of cell you isolate. And we in the lab, I'll not go in the detail about the amplification method we use, but when I started my lab, we use this MDA multiple displacement amplification. And now we are doing the primary template amplification. And what we see that in a very high DIN, I'm showing you an example of a very high DIN as well as a low DIN and a 2 different amplification method. And how we read this kayak cell output is that, so this is an empty well, the upper boundary and lower boundary. And we are looking for this amplification of this 4 band, which expands from 140 to about 540 base pairs. And we are looking for this, this 4 bands here. And what you see that with the MDA for the, so that these are the 2 different cases, but the DIN is very same. And what we see that when we do our MDA amplification, we can see that 3 or 4 of those chromosomes are amplified, but not in every cells. But with the PTA, we have higher success with the amplification. And it looks like, this DIN is not associated, we see the DIN is not associated with like, what is the amplification rate? It's our amplification method is associated. So you could have a low DIN, which is 5.8 here, and we could still get, perfect cells. And again, we are not going to amplify hundreds of cells. We are going to amplify 10 cells or 15. We are going to sequence 10 or 15 cells. So this is perfect for us. We could select some of the good amplified nuclei from here to our further studies. But again, as I said, it's a very expensive method to do a sequencing of whole genome. So we add a second layer on top of this. Once we see that these cells have amplified evenly, we choose the cells from here. And by MDA, we get about 10 and 15% cells where we have good amplification. And for PTA, we get about above 85% or 90% of the cells get amplified evenly. But this is our first quality check for the whole genome amplification. But later we do another, when we used to do MDA and our success rate was not so high, we also did a very low, like 0.5x whole genome amplification of those selected nuclei which had more than 3 bands by our multiplex PCR. And what we are showing here that this is the MAPD score. This is the median absolute pairwise difference. This is actually for microarrays was used, but for our study, we use this as a quality control. And if the MAPD score is low, it's considered to be good because then you don't see your chromosome to chromosome variability. So we are seeing that how evenly each chromosome has amplified in this single nuclei. And it could vary because, it depends on your study. So it's possible that, your MAPD score is all above 1, but in your all cells, if you see, you can you can set up a threshold. For this study, we put our threshold 1.2 and we were only sequencing 3 cells. So we got even with the good DIN or even with a very low DIN, it looks like we can get few cells even with the low DIN tissue. And we were very happy with this. But with the PTA, our life has recently changed where we could amplify most of the genome, and we are not doing anymore this 0.5x whole genome amplification. So this is, that is what I showed you about our whole genome DNA amplification. For this RNA from this tissue, we again did the RNA-seq, 10x RNA-seq from a high DIN tissue as well as a low DIN tissue from our sonic method. And what I show you here that in both the cases, we could get not all the 13 cell type, but majority of those cell type we could capture by this RNA-seq from this both of the tissue. And what we also did that we took this tetraploid population and we did the RNA-seq, and what we see that this tetraploid population above 98% of those cells were actually cardiomyocytes, and very few was endothelial or the fibroblast population. So what it showed us that, this tetraploid or higher ploidy population is a pure cardiomyocyte population in case you are interested to, look at the changes in the genome, only cardiomyocytes, because again, cardiomyocytes don't have very good marker for that. So for that, you can use this higher ploidy population, and human heart, as with increased age or with increased disease, will have a huge population of the higher ploidy cardiomyocytes. And at this population is again might be capturing the changes in the genome for the disease or for the aging. So this was a very great way to identify the cardiomyocyte population in absence of marker, antibody marker. And this is not in the paper, but I just wanted to show you what you can do with this method. We had isolated using this sonic method, and we did the single cell whole genome amplification. And this is from my other published article, where we show that with the increased age, we see the increase in the somatic mutational burden in the human heart. And this is this plot actually indicating that all these mutations are actually spread evenly over this genome. And we were also able to identify this different substitutional type in the human heart. And the substitutions are color-coded here. And we see that in the heart, many of those mutations are coming from C to T substitution or T to C substitution. And we did this relative contribution of this substitution. And with this, we found that in the human heart, we have 3 different types of mutational signature. And from the cosmic, next we compared this, our signature, and we find that this signature A, which has been known as the aging signature and is present in most of the tissue, and it's very close to this SBS5, where is mostly T2C or C2T mutation. And as you know, and again, this amplification was done in tetraploid. So this was wonderful way to prove that, our sonic method is working and we are capturing the true signature of the mutational signature, because all of our cells had this SBS5 signature, which is the aging signature. And we see that with increased age, we have this increase of these mutations, and many of them are coming from this signature A. And more interestingly, as that we find this signature C, which is actually this signature A, matches with the SBS signature 8 and 18. And interestingly, this is actually reactive oxygen species mediated mutational signature. And as you know, heart is a very highly metabolic organ, and there is a huge oxidative demand. And with increased age, we see this oxidative damage, and this is mostly C2A damage. And in the heart, we see that damage, which is very, again, reassuring that, we are isolating, amplifying, and capturing the true signature of these cardiomyocyte mutations. And then we were also able to identify another signature, which is called the SBS44, and here as signature D, which actually shows that the defective DNA repair mechanism in this. So and this is, I'm not going to get into the detail of that study, but this is also the only one cardiomyocyte specific signature and not has been seen in any other tissue. So we were very glad that sonic method had worked well, and we could isolate the cardiac myocyte nuclei very effectively and capture the true changes in their genome with the aging. And what our method actually is showing, that the highlights of our method, that this is the ideal method that you could get the cardiac nuclei for whole genome amplification and the RNA-seq. And then if you are doing, and this ploidy best, the single nuclei sorting could give you the pure cardiomyocyte population if you only want to look at the cardiomyocyte genome or cardiomyocyte transcriptome. And then, we would really suggest the lab to not only focus on the RIN, but also at the DNA integrity if you are going to work on the DNA of that from the tissue. And then we would like to say that, if you have DIN over 5.8, it will be like, you could still capture most of the changes in the genome on the transcriptome. But the drawback of this study that, all the tissue we collected had the RIN all over 7, but we were able to get one tissue which was not so ideal for our study. But even our method shows that we could get, if you do not need a lot of population, and you could get some population and you could capture the changes in that genome and the transcriptome. But you have to keep in mind that, it's possible that during that aging or the disease progression, that the cell has damage to that point that if you only collect the best nuclei, best amplified, you might not be, capturing the biology. So you have to understand like what you want to do and what you want to capture. And so with that, I would like to thank my team. And as this was a very joint collaboration between the translational research team to get the tissue and do the QC of the tissue. And I am so thankful to Sarah to put this together and everybody in our team. And this is like, this sonic method is our bread and butter. Our week starts with that, our day start with that. And we feel that this is a very good method to capture the DNA, RNA changes in genome and the transcriptome. And I'm thankful to Ron, who has worked tirelessly to get this right population for our study. And I'm thankful for all the donors who have donated their body, their organ for this research so that we can understand that the genome and transcriptome changes, and we could do all this wonderful discovery. And last but not least, I'm thankful for all this, all the funding agencies to support our research. And with that, if you have any question, I'll be glad to take that. And I'm going to stop sharing so that I can see your faces and I can talk. And but if you need any slide, I'll go back from there. And thank you so much for your attention and time.
Thank you, Sangita. That was really interesting and informative. And I do some transcriptomics and epigenomics from human tissue as well. So I have some questions. There's a few in the chat already or in the Q&A. So while people are maybe thinking of some more questions, I can start with a couple.
Yeah, sure.
So, I'm somebody who I guess a lot of people are, who if I have some human heart tissue, like you say, you want to make sure that how the quality is. I look at the RIN and then I still isolate RNA and DNA and assume the DNA is going to be fine if the RNA is. So I was wondering, maybe I missed it. Did you correlate RIN with DIN, and was there a correlation?
I did not do the RIN and DIN correlation, but it could be very different. If you look at the paper, you could, so it depends tissue to tissue, but sometimes we see the RIN and DIN was like all at the similar level. But if you look like hundreds of tissue, I gave the RIN and DIN on the paper, and there you could see that the DIN was like perfect over 9 and the RIN was like 5.8. So then I was like, should I use this tissue or not use this tissue? And it will so much matter, like, what you want to do in this study. So it's very important that, if you are going to do the DNA work, check the DIN for that, because it could vary. But most of the cases, it was pretty similar. Like, if the RIN was above 7, the DIN was also above 7. But some cases we see this variability.
Yeah, very interesting. I also wouldn't have thought to look at the DNA integrity. Yeah. Okay, so we have some questions. So if it's okay, I'll read them out and then you can answer them. So the first one from Sanjiv Shah is, approximately how many nuclei are you able to isolate from the smallest amount of tissue, for example, 10 milligrams that you might get from a biobank?
You can get above, if we have very little tissue, we start with 10 milligram and we could get like 1,000 nucleus, like the whole nucleus from. But then if you are looking for cell-specific way, then you have to remember within this 10 milligram, you have to have your control. where you have no stain, or like, to set your gate. So it depends like what you want to do that. If you just want to do RNA-seq and you are taking all nucleus, you will get, couple of thousands nuclei from there. But for me, if I'm going to look for my tetraploid population, I might get, maybe 50, but I'm good enough because I don't need more than 50. But yes, you have plenty, even from the 10 milligram tissue to start with, make your control to set your gate, and then, have your stain population or your, your wanted population in there.
Thanks. Yeah, it makes sense. A question from, forgive my pronunciation, Rajiven Shri, I'm sorry, I can't pronounce the surname, asks, great question. Did you also adapt your protocol for formalin fixed tissue?
So we are trying. So this is, it's a great question, because most of the tissue repository, not most, many of the hospital tissue repository, they don't do a fresh frozen. They keep actually formalin fixed. So our lab is actually working to see that if we can do the whole genome amplification from this. And there are few articles actually, they show that you can do the amplification, but again, what do you want to look? Because that DNA is like, the formalin got entangled with the DNA. So how are you going to like, how are you going to actually integrate that result into your data? Are you going to look all formalin fixed, your disease and your control, and how long they have been fixed? So I am not very happy. You can use this method, but then depends what you want to do it. Is this the only method? So in that case, you will go, if you are going to look at the transcriptome, then there are like MERSCOPE and VZM and all this. That will be the better way to do it than the single cell like RNA-seq or the DNA-seq on that, because you could also from those VZM or nanostring or MERSCOPE, you could still identify the mutations and the changes in transcriptome, but not for the MDA or PTA. Those amplification does not work. So like even if it works, you do not know like how well the genome has amplified and how well the transcriptome. So I will say use with caution if you have only formalin fixed tissue. But we are working in the lab to see like when you cannot get your DNA back, because we know if we like we are very like if we do a 10 minutes fixation, we can get amplification with MDA and PTA. But when we get from the tissue repository, which I have no idea how long they have fixed, we cannot get any DNA, single cell DNA from there.
Yeah. Next question from Priya Gura asks, did you see any changes in gene expression between diploid and tetraploid cardiomyocyte nuclei?
Yes, it's coming. You have to just wait. I cannot give up yet, but yes, our lab is working on the changes with the ploidy and in the genome. And of course, we do see the changes, but I will not spoil. Then my postdocs are going to be really mad at me. So because this is their project now to see the changes in the genome and transcriptome with the ploidy in the heart tissue. Yes.
Yeah. Okay.
Very good questions. Yeah.
Honor Rhonda asks, does DAPI affect the DNA quality in any way? And can you recommend any alternative DNA stains if this is the case?
So for the so some studies, like I think for the nanostring, when we were working with them, they didn't want us to use DAPI, and but for the whole genome amplification, the way that for the cardiomyocytes especially, and then there are so much debris, and we want the nucleus, so it was how to say, again, a balance to use or not to use the DAPI. And a lot of studies has been done in the whole genome amplification with the DAPI, and it has been shown actually that it does not changes because again, these cells are frozen, and if we go to the facts, they will say it's a dead cell for you. But for us, even if it's dead, we can still capture. So this DAPI did not actually interfere after the tissue was frozen on that case. That's what I would say. And I have not done any QC, non-DAPI sorted versus a DAPI sorted. So I cannot tell it much, but, I know that some researcher from our division actually did that, and they did not see much changes with DAPI and without DAPI sorted, changes in the genome. But again, when you are doing in single cell, each cell, we see so much heterogeneity that, you do not know if you're just lucky, on that day that your 5 cells, shows DAPI positive and DAPI stain and non-DAPI stain was like perfect or not. But we don't see that much changes with the DAPI stain.
Okay, thank you. Rajiv then asks, this is the person that asked about the formalin fixation earlier, says, thank you for the amazing talk. And did you look at SNPs and other mutations in the aging hearts of patients with congenital heart defects?
Our study was with the only aging with no cardiovascular disease. And we are very much interested now to look at the changes in the genome in the somatic mutation SNPs. So we will look into that. But with the aging, we also looked into the small insertion and deletion. But we were not very confident about, that our algorithm was correct. So we have not included in this. But now with the new amplification method, we are very confident that, our insertion and deletion, what we are looking is true insertion and deletion. So yes, with the aging, you do see somatic mutation is not only single nucleotide, you have insertion, deletion, you have retrotransposition, you have inversion. So our lab recently just got into this consortium. It's called Somatic Mutation Across Human Tissue, SMART. And there actually we are looking all types of somatic mutation, not only single nucleotide changes. And this is all with the non-diseased tissue across all the tissues in human body. So I think a lot of data is coming up. Finally, NIH is invested to look at the somatic changes in the genome, not only hereditary changes in there. So I think, in next 10 years, you will see, all of this coming up, and, stay tuned to that.
Thank you. Excellent. Another question from Jasmina Kvetowska. Says, again, great talk. And what's the time frame that tissue can be used after snap freezing for your protocol? And regards from Sydney, Australia.
Oh, yeah. Thank you so much, Jasmine. Yes, this is also, I really want to know how long the tissue has been frozen, but because I'm getting it from a bank, I was thinking when actually I was collecting, to ask, when did you collect this? But I was already asking so many questions. But I would say I have worked recently with some of the tissue, which I definitely know has been stored, snap frozen, over 20 years. And we did the RNA-seq from there. And we do see that, the tissue which I have collected, after I started my lab in 2 and 3 years. But of course, I'm when I am freezing, I'm doing for isopentene, and I am doing slow, I'm making sure I freeze properly. And I do not know how 20 years before it was frozen and what was the postmortem interval. And after coming to lab, how many hours it was sitting on the ice. So I do see, and then again, the one which were very frozen 20 years before, they're also diseased tissue, not control. So not disease, but I do see that I get less number of nuclei from a very old frozen tissue. But again, those are disease. It's comparing this postmortem tissue is so much harder than we think. And when you are interpreting any data from there, it's very important to take all these covariates, how you are going to interpret. So yes, I would say I do see changes, but because I also collected disease tissue and very bad disease, heart failure tissue, and I will still get much more number of the nuclei, which I could do successful amplification versus disease tissue, which was stored 20 years before And now I am doing amplification from there. So I would say that, although I don't have evidence, but from my small, experiment between like 7 and 10 tissues, I have noticed that.
Yeah. Okay, thank you very much. So the tissues that you've had from the bank, were they all post-mortem or the disease from explanted hearts? Do you know?
So if it was control, that means the control I defined as they died of car accident.
Yeah.
And again, in the neurobiobank, because, they get the whole body most of the time, and the explant is very rare. So those are all postmortem. Then, like in this study for this JMCC, all tissues were collected from the neurobiobank. So they're all postmortem. But now I am working on the disease where I'm getting like explanted or surgical discarded, where I will have a different tissue quality. And I think I have to come back in 5 years and update our community, what I find from there.
Yeah, awesome. We've got a few more minutes. I'm going to be very selfish and ask another question for my own interest.
Sure.
So the digestion of the frozen tissue, I think it was 37 degrees for 10 minutes. Mm-hmm. Did you put inhibitors of RNA polymerase or of epigenetic modifiers in there to stop changes occurring during isolation?
Yes, good, very good question. It will be, so if you are going to use that, so the first thing, from this postmortem tissue which are frozen, if I am going to do anything more than just staining, I would put lots of RNase inhibitor or whatever you need if you are going to do this one, because that the moment you are defrosting, the tissue is started degrading, right? So in this method, I was just staining, so I did not use any inhibitor, but the moment I do my DNA or the RNA work, the moment the tissue gets the buffer, I load with the RNase inhibitor on there. Yes.
Okay. Excellent. Thank you so much.
Mm-hmm.
I think all the questions in the Q&A have been answered. Mm-hmm. So if Ron doesn't have anything else to add, then I'll just say thank you so much, Sangita. That was really interesting and very applicable, to work lots of people are doing. Mm-hmm. On the human heart. So thank you so much, and look out for this webinar going onto YouTube soon as well.
Yeah, thank you so much, Emma, and thank you so much everybody, for their time and the wonderful comment. And stay tuned for what's coming from our lab.
Awesome. See you soon. Thank you for
everybody. Bye. Take care.